So today we are going to be looking at the fermentation abilities of our microbes. So we're going to be testing the fermentation ability for 3 different sugars. We have one tube that contains lactose. We have one tube that contains sucrose. And we have one tube that contains glucose. Our lactose and sucrose are going to be broth cultures. So basically what we're looking for here is a color change. Our media contains phenol red. Phenol red is a pH indicator dye. It is red at a neutral pH, and it will change to a yellow color in an acidic pH. So for our lactose broth, we are going to be testing for the ability of your microbe to ferment lactose. So it can use lactose as a carbon source. The lactose broth is the one that contains the white plastic cap. You will notice that our lactose broth contains a Durham tube. The Durham tube is that inverted vial. It is filled with liquid, and it will capture any gases that might be produced by your organism. So for our fermentation experiment, we can have 3 different possibilities for our results. We can have an organism that is positive for acid. That means that fermentation has taken place. We can have an organism that is, positive for acid and positive for gas. We can have an organism that is positive for acid and negative for gas. And we can have an organism that is negative for acid, which means that it has to be negative for gas. You can only have gas production if your organism is positive for acid. So we are going to inoculate our lactose broth. So you just sterilize your loop the same way you normally would. You will then pick up one isolated colony from the plate, and then you're going to use that to inoculate your broth. So we're going to inoculate our lactose broth. Just be careful that you don't move the Durham tube around too much. And then when you are done inoculating your culture, do not vortex. We don't want to break the Durham tube, and we also don't want the Durham tube to lift up off the bottom of the test tube. So now that we've inoculated lactose, we're going to inoculate our sucrose broth. So our sucrose broth is basically the same as our lactose broth, except it contains sucrose instead of lactose. It also contains the pH indicator dye phenol red. So we're going to do the same thing. We sterilize our loop. We will pick up a colony from our plate, and then we will do our inoculation. So again, we have 3 possibilities here for our results. We can have an organism that is positive for acid and positive for acid and positive for gas. We can have one that is positive for acid and negative for gas. And we can have one that is negative for acid and negative for gas. The last fermentation that we're going to be looking at is for glucose fermentation. Now, our glucose fermentation tube is going to look a little bit different. This one is actually an auger, and it has been made into a slant. So our glucose iron auger is going to contain the sugar glucose. So we're going to look at glucose fermentation. It also contains iron salts. Iron salts are going to tell us if hydrogen sulfide production is taking place. So we're going to be looking for 3 things in our glucose iron auger tube. We're going to look for glucose fermentation, we're going to look for hydrogen sulfide production, and we're going to be looking for the oxygen requirement of our organism. So depending on where your organism grows in this tube, we can tell what our oxygen requirement is. And again, glucose iron auger contains phenol red. So to do our inoculation, we're going to do something called a stab inoculation. With a stab inoculation, you are going to be using an inoculating needle. An inoculating needle looks basically like a loop, except it doesn't have the loop. It's just the wire. So it works the same way. You sterilize your inoculating needle, and then you're going to pick up a colony from your plate. And then you're gonna do the stab inoculation. So to do a stab inoculation, you are going to take your inoculating needle, you are going to insert the needle into the auger, and you're gonna stab down to about 1 centimeter from the bottom of the tube. You then pull the needle up along the same stab line, and then the last part is to do streaking across the surface of the slant. So you just move your needle back and forth across the surface of your slant to streak it out, like so. And that is how you inoculate a slant.